I think the challenge was always having enough money. I went without salary for a few months. Both Ken and I lent the company a significant amount of money to make sure everybody had salary. There were times when it was quite painful, but in the end, we were lucky enough to pull through. And when we were eventually acquired, I think one of the highlights of my professional career was bringing people in at Specifica and telling them what they were going to receive as a result of the acquisition. Everybody in the company, from the lowest to the highest, had shares in the company. It was really quite a high point to be able to tell people. Some of them were getting life-changing amounts of money. So that was really very exciting.
Wow. Most people will never meet the person who trained under the man who invented the antibody drug. Today you will.
Andrew Bradbury spent 7 months without a paycheck keeping his company alive and then built the test the entire AI biotech industry was afraid to run.
This is Authentic Conversations. Real stories, true talk, straight up. Welcome. I'm Deanna Brown.
And I'm Jeffery Davis. Today we're talking to Andrew Bradbury in Santa Fe by way of Cambridge, Italy, in a national laboratory.
Andrew founded Specifica, an antibody discovery company that now supplies most of the world's largest drug makers. But the part we're here for isn't the acquisition headline. It's the 7 months. It's the bet he made on the unproven idea and the uncomfortable questions he's been willing to ask out loud.
If you've ever been sold a technology where it was going to change everything, this one is for you. Andrew, welcome.
I think I feel a bit embarrassed, to tell you the truth, by that introduction, but never mind.
That's part of the fun here, Andrew. Andrew, you trained under César Milstein, the person who invented the monoclonal antibodies. What did being in that room early teach you that shaped everything thereafter?
So César was relentlessly focused, I would say. He actually had a lot of scientific discoveries to his credit. The invention of monoclonal antibodies, or the discovery of monoclonal antibodies, was one of them. And he saw the commercial potential for monoclonal antibodies and, in fact, brought them to the attention of Medical Research Council, who did not see any commercial value. And so he published it. Now, what he did was made mouse monoclonal antibodies. So although initial experiments injected these into humans, they could not really be used. The antibodies needed to be made more human. And that basically was the start of a whole industry, really, leading to human antibody therapy, which is a multi-billion, hundreds of billions of dollars a year industry.
It's fascinating to me that they didn't see any commercial viability in something that seems like it would be so revolutionary in the health and wellness of mankind.
I think in retrospect, everything's easy. It's just at the time, and I don't think the people making the decisions were scientists or physicians. And at the time, it was not seen as something that was going to be. Well, all the people all the people around César thought that it would or could be very valuable, and I think it was the people in head office of the Medical Research Council that didn't see it.
Andrew, can I ask you, for everyone outside of biotech, because as we talked about, we have a pretty broad community of entrepreneurs and really wonderful and smart people, but we're not all biotechnicians. Explain the mice versus libraries sort of thing here that we're talking about, and how these libraries or even the mice kind of fuel the industry.
So let's go back a step and talk about antibodies. We all generate antibodies in our cells, and antibodies are what help us fight infections, okay? And so, for example, vaccines, when you get immunized with something, you will develop an antibody response to the vaccine, which mimics the pathogen that you're trying to protect against. And so you get an antibody response, the pathogen infects you, and now the antibodies bind to the pathogen and stop it causing further damage. So an antibody is quite a complex molecule. It's quite large. It has two bits, basically. On one end, you have what's called the binding end, the variable region. And there are trillions and trillions and trillions of different binding regions. Each individual antibody will bind to a different target. The other end of the antibody is a constant region, and the constant region is responsible for providing the biological activity to the antibody. So once an antibody latches onto a target, it can induce a whole series of different biological responses. If it's binding to a cancer cell, it can kill it using various mechanisms. If it's binding to a pathogen like a virus or a bacteria, it can cause phagocytosis. And so that's the concept of antibodies. And antibodies were seen as magic bullets. There was this idea that if you could get an antibody against a cancer target, then that would be a magic bullet that would kill the cancer. Now, the first antibodies César developed were mouse antibodies. And trouble with mouse antibodies is that when you inject them into people, into humans, you get an anti-mouse antibody response. It's a human anti-mouse antibody response. And so, although there were some mouse antibodies that were approved and were used, now the field has moved completely over to human or humanized antibodies. Human antibodies are ones that really either come from humans or from transgenic mice. So let me go back a step. If you immunize a mouse, it has a whole bunch of mouse antibody genes in it, and it will respond with a mouse antibody response. And so to make what are called transgenic mice, what they do is they remove all the mouse genes and they replace them with human antibody genes. So now, when you inject a target, a pathogen, whatever, into the mouse, instead of responding with mouse antibodies, it responds with human antibodies. And in that way, you can get human antibodies by immunizing a mouse. So that's one way of getting human antibodies. Another way is you immunize a standard mouse. You get an antibody, which is a mouse antibody. So these are like the antibodies that César developed. And then you look at that mouse sequence, the mouse antibody, and you change various bits along the mouse sequence to make them look more human.
Okay.
That's another way. That's called humanization. And then the third way of making antibodies is what we've been helping develop, and that is you make what's called a library of antibodies. What you do is you create what's called a library of human antibodies in a tube. And so a tube, something as small as my finger, may contain 10 billion or 100 billion different antibodies. And so how do you get antibodies from that is you imagine this is the tube, you put your target in, and this is all metaphorical. Put your target in, you mix it around, you pull it out, and then antibodies will be bound to that target. And now you isolate the antibodies that are binding to the target. And it's more complicated than that. So this technology was developed, in fact, in the same place where I did my MRC by a scientist works in the lab opposite mine. He was called Greg Winter. He also got the Nobel Prize for this technology. That was done in 1991. And the promise at the time was that in vitro antibodies or antibody libraries were going to replace the immunization of mice. That was 35 years ago. It hasn't happened. The market is about a third human libraries, third transgenic mice, and a third humanization of traditional antibodies. So it's continuing. Now we've got a new player on the block, which is AI, and there are a few examples of AI helping in developing antibodies. But I suspect that in the long run, AI will be yet another way of getting antibodies alongside the other ways.
Got it.
Fascinating.
Yeah. I want to drop into another level of the creation and the production of this, and maybe even just step back a little bit. When you stepped into this very large endeavor, and you essentially, you and your wife made a decision to wait till when the kids left before you started the company because you knew it was going to be a large endeavor. Walk us through that decision. Did you see the potential for what you were doing, or were you just kind of, were you betting the farm, so to speak?
So I'd always wanted to set up a biotech company, and my wife had always said, let's be careful about this. And she was the one that really said, let's wait. And then when the kids left, she said, okay, now you can do it if you want. And as it happened, our business colleague, fellow called Ken Sharples, who co-founded the company with me, he had just come off another venture. He approached me and said, well, you interested in doing this? And I said, yes, why not? I continued to work at the National Lab while I was a consultant for the company for the first year. And then after a year, I realized that this really wasn't going to go in unless I jumped full in. And I was very excited to do so. And so that happened in 2017.
In the process of working on something large and creating something, there's never a straight line. Was there a spot where you hit a wall and you were trying to problem solve your way through? Take us into a moment in the development.
So, right. So we were lucky. We had a couple of small contract right from the very beginning, and we developed another one quite soon after. And so we had decided to bootstrap this. Ken had been involved in venture-funded biotech companies in the past, and he had pushed to do this bootstrapping. I was really a bit naive at that point. I just took his advice. He was the business guy. And so we had thought that we would develop an antibody discovery company. There were quite a few others out there. I also had felt that there was a way of making better antibody libraries. And so it was after a couple of years that we were able to do that. And the new library design that we made, which we were able to patent, really formed the basis for the company. I mean, there's a famous story about Boeing where when they developed the 747, they bet the whole company on it. And they weren't sure whether the 747 was going to be successful or not. And if it failed, well, that was going to be the end of Boeing. We were a small startup there. But in the same way, I felt that if this hadn't worked, if this technology hadn't worked, then I think we probably would have been toast. There are a lot of other companies out there doing antibody discovery, as I've said. And I think what we now are able to bring is a library design which gives you really high quality antibodies, and that's why about 50% of the top 20 pharma have taken our platforms in house.
Just to further that thought, you bet the company on this new library format. Why were you certain? Was it just instinct, or was there some data to support that?
So I wasn't. I think it's instinct more than anything else. There were other things that had been done that were similar, but I felt hadn't been done as well as they could have been done, which is the approach we took. And I think it was just instinct, a feeling that if we did this properly, it could work really well. But we were surprised by the results. It worked actually much better than we were expecting.
The thing that pops to mind is the fuel for the creation of something so significant for you seems like it was two things. One, the fire behind you was no plan B. And two, the guiding light was inspiration, because you were following a gut instinct. I'm saying those were part of the process. Everything obviously was data-driven and work-driven and everything else. But without those two things, it might have been very difficult. Do you think that that's a fair assessment of what was helpful along the way?
So it would be nice to sort of claim that I had a big vision and all the rest of it. I mean, to a certain extent, we were sort of struggling to survive, and part of that struggle was developing a new product that I thought would be differentiated from what everybody else was offering. But we really didn't know until we'd actually made the libraries and tested them and saw how good they were. There was an instinct, certainly, that this could be better, but there are many examples of other people in the field who have developed libraries that have turned out to be okay, but nothing really special.
So it was really your approach and ultimately the diligence or the discipline around the methodology that sort of differentiated your approach versus others, or
so
is there something more scientific?
So let's have another discussion about antibodies. So when you have a therapeutic antibody, this is something that you inject into somebody, and you either inject directly into the veins or you direct under the skin, and you inject large amounts of that antibody. Now, that antibody has to have three properties. One is it has to function. It has to bind to a target and do something. Thing is, when it binds, it has to have a certain affinity. Usually, you want it to bind with a high affinity, which means it binds very, very tightly and doesn't come off. And then the third thing is a concept called developability. And developability is this idea that what it takes for an antibody to be developed into a drug. So it's not enough to have an antibody that binds if you can't make it, or if it binds and it has an activity, but then you inject it into the bloodstream and it induces an anti-antibody response, or it aggregates, or it has an off-target effect, it binds to something else. And so all of these properties are called developability. And with our libraries right at the very beginning, we were trying to solve this developability problem. There were
Got it.
sequences in antibodies known to create potential problems. And so we created a library without any of these sequences at all. We eliminated essentially all of them. What we found was, surprisingly, is that in addition to solving this developability problem, we also, to a certain extent, solved the affinity problem. So the antibodies coming out of our library were much, much tighter binders than coming out of other libraries, and indeed libraries that I had made in the past as well.
Fascinating. All right, that helps a fair amount. You mentioned AI and how it swept into the antibody discovery business. A lot of money, obviously a lot of hype. And tell us a little bit more about this competition you created, and handed it off to a nonprofit just to keep it safe and not attached. So tell me a little bit about that story.
Okay, so AI jumped into the antibody discovery field just like it's jumped into pretty much everything. And that was about 3 years ago. And I regularly go to conferences, and what I started hearing at these conferences was AI was doing this, it was doing that, it was going to solve everything. And essentially, the feeling I got was that AI was really going to eat everybody's lunch. There wasn't going to be any room for experimentalists. The only role for experimentalists was to really just test the outputs of the AI. Now, I have been involved. I've lived through quite a few hype cycles in antibody discovery, and I'll be honest, I've been responsible for hyping a few technologies myself, including phage display, which was when I started. Um, and I just felt, well, if it's really that good, let's find out. And so we at Specifica developed a set of we'd done some selections. We generated large numbers of antibodies against COVID, and these databases of antibody sequences against COVID were not useful from a commercial point of view. And so what I thought was, well, let's design a series of challenges based on these data sets that we've developed against COVID, and see if those companies and institutions, academic institutions using AI in antibody discovery, see what they can do with it. The rationale behind using COVID, apart from the fact that it was not useful commercially, was also COVID and the binding protein of COVID is the most studied protein in history now as a result of the pandemic. And so the feeling was, if AI could not solve these problems with COVID, it was going to struggle to do it elsewhere. And so we published a manuscript in the fall of 2024, basically announcing the competition, describing what we were going to provide everybody. And then at the beginning of 2025, we basically allowed potential participants to download the data sets all simultaneously. And then they had 2 weeks to fulfill the 3 competitions within the challenge that we had set them. And then at the end of 2 weeks, they provided the sequences of the antibodies that they had designed or identified. I had arranged a consortium of companies to produce those antibodies, and then to analyze them both in terms of their binding activity, in terms of their developability, which is that concept I mentioned before, how tightly they're bound. And we were able to analyze, eventually it was over 500 different antibodies provided by 29 different organizations. Now, this was the state of the art in Q1 of 2025 for those 29 organizations. I want to stress that caveat. But first, I think there was no AI algorithm that was useful in all 3 competitions. The second was that there were some algorithms that were better for one thing than another. And for one of the particular challenges where we'd asked the participants to identify high affinity antibodies, you were actually better off for all but one algorithm just picking clones randomly rather than using AI to help you pick your clones. So the paper describing the results is now in press. It should be out in a couple of weeks. But I would say that on the whole, there are places where you can see AI may be helpful. But certainly at the moment, and under the things that we tested, it looks like there was quite a bit of hype there. You asked about the competition and passing it over to nonprofit. So
Yeah.
so when I had this idea for a competition, I spoke to a few other people in the field, and some of them wanted to set up a consortium with big pharma, academics, and my feeling was this is going to take forever. It's going to take 3 years minimum, and the field is
Yeah.
moving so fast. We need to get on and do this as fast as possible. And so that's why we did it within the context of the company. There were advantages to that. We could do it quickly. Disadvantages, it's a company, and so people are naturally suspicious.
Yeah.
And so that's why we've handed it over to the Antibody Society, which will look after the next phase of the competition.
That's a wild story. That's fascinating. I guess I'd ask you the big question over the course of these two startups. What were the highlights of those years, and or as well as what were some of the challenges? If you had just to broad strokes tell us about your human journey through this process.
So, I mean, in terms of the startup, I think the challenge was always having enough money. And I went without salary for a few months. Both Ken and I lent the company a significant amount of money to make sure everybody had salary. That there were times when it was it was quite painful, but in the end we were lucky enough to pull through. I think the acquisition was Ken had always wanted us to be acquired, and so he started the acquisition process. And when we were eventually acquired, I think one of the highlights of my professional career was bringing people in at Specifica and telling them what they were going to receive as a result of the acquisition. Everybody in the company, from the lowest to the highest, had shares in the company. It was really quite a high point to be able to tell people. Some of them were getting life-changing amounts of money. So that was really very exciting.
It's exciting. So I do have a question. You're on the leading edge of this. What you're doing is shaping some of what can be done and what future things will be built on it. Are there things that the insiders understand and discuss privately that the public rarely gets to hear?
Well, I think the public becomes aware when they see adverts on TV saying, cure your psoriasis by getting this drug. And you can always tell if it's an antibody drug because it has MAB at the end. You'll have the company name for the drug in large letters, and then underneath there's what's called the generic name, and that is always something, and then MAB at the end, rituximab, for example. And so you can always tell if it's an antibody. And most of the adverts on TV are, in fact, for antibody drugs. Now, I think what the public doesn't really probably understand is just how much work goes into developing these drugs. There is a tremendous amount of work. You start off with a hypothesis. You test that hypothesis first in animals, in mice. Then you move to monkeys, and then eventually you test them in humans. First test is to make sure that the drugs do no harm, and then later tests will see if they have any efficacy. And I think that what's going on is this is based on a very careful desire to ensure that drugs do no harm, and it's important that I think drugs are tested and developed in this way. The world is full of charlatans that are very happy to sell snake oil. And I think if a drug has gone through the approval process, then usually it will be functional and it will have relatively few side effects, although the side effects are often known. And in those adverts, you will often hear a long list of potential side effects at the end. Now, that's not to say that drug companies don't try and develop drugs that have marginal benefit over what already exists. But nevertheless, every drug out there pretty much has undergone rigorous testing, and I think we should be grateful for that. Otherwise, what happened in the UK with thalidomide, which for those of you I don't know who may not remember, it was a drug given to women to, I think, in pregnancy, I think to help with headaches, I think. The result was that kids born to mothers with taken thalidomide ended up with missing limbs and things like that. And it was partly as a result of that the US developed this very rigorous drug development and assessment process.
I did not.
Andrew, we asked you a handful of questions. What's the one question that you wish people asked you more?
I guess one question is, would you have gone into biotech earlier if you could have done? And
Yeah.
I've enjoyed, notwithstanding all the difficulties, I've really enjoyed setting up a startup. It's been really challenging, but and it's been painful and it's been difficult, but it makes you feel alive, and that's quite exciting.
Yeah.
Now, I think my wife was right. I think we were incredibly lucky in the time when we came out. COVID came along. There was a massive investment boom. After COVID, we were acquired just after COVID. I think we were incredibly fortunate in everything that happened. I'm not sure we would have been equally fortunate had we done this earlier or even later. But nevertheless, doing a startup is really exciting, and I recommend everybody trying it.
Absolutely. Well, as two fellow entrepreneurs with you today, I would agree with you.
Absolutely.
I'm on my 6th startup at this point, so I'll let you know how it goes.
Congratulations.
Thank you very much.
Andrew, thank you. And this was exactly the kind of conversation we set out to have with you.
It was insightful. I have to admit, I learned a lot about biotech and antibodies for sure. I think I can go back and take that 11th grade science class head on at this point. But it was fascinating. I also really appreciate your human story. And so we're thankful for that. We're going to say goodbye now. And
Thanks very much.
To our listeners, come back for another episode of Authentic Conversations. Real stories, true talk, told straight. Take care.
Bye-bye.
Thank you, Andrew. All right.
Thank
Bye.
you.